Hello everyone, my name is Aditya Mahesh and today we're talking about CRISPR-associated proteins, specifically SpCas9. But before I get into all the details explaining you what all these colors and all that mean, let me talk about the CRISPR systems. They are split into two groups. There's class 1 and there's class 2. Class 1 is a combination of many proteins that make the gene editing happen. And in class 2, it is just one big protein that does all the work. And in class 2, there are many different subtypes. There's 3. We're just going to be focusing on the first one, which is type 2. And this specific Cas9 protein is involved in the first subtype, or type 2A. Here you can see the many steps inside of a CRISPR system. After a virus injects its DNA, the DNA goes through the first step, spacer acquisition. Many proteins cut out a protospacer from that inserted gene, incorporates it into the bacterial genome. And then you have CRISPR biogenesis. RNA is processed. Here we have this RNase 3 Cas9. The big part that we're all interested in is the interference. And in here, we're using Cas9 to edit the gene. Let's get into the Cas9 structure, the type 2A Cas9. So in here, you can see many different colors, which represent many different domains. This specific Cas9 has 1,368 amino acids, and this Cas9 participates in CRISPR RNA maturation, and it also has to do with CRISPR interference. SpCas9 is an enzyme, and it is activated by a coenzyme being the guide RNA. The model came with a single guide RNA inside of it. Cas9 is bilobe, meaning it has two main lobes. It has an alpha helical recognition domain, that is what you see in gray, and then you have a nuclease lobe, that is all the other colorful parts that do a lot of the cutting functions. These two lobes are connected with two linkers. The first one is the arginine-rich bridge helix, the violet-purple segment that you see in here that connects the two lobes, and there's also a disordered linker down inside here that's yellow in color. The rect domain is made of helices, and in this protein there's 3 main helices involved. Nuc domain has the H&H domain, and this has to do with making a cut in the target strand. As you can see here, that's right here, and it's with the orange target strands. This H&H domain is very conserved, meaning over evolutionary scales, this really hasn't changed in many different species. And this is similar for the RUVC domain, which cuts the non-target strand. This is split into many subdomains. I've color-coded it all the same, but there's actually 3 different sections. You can see that in the sequence up here. There's 1 up here, the blue, there's 2 more down. And finally, there is a variable C-terminal domain. The C-terminal domain has a Cas9 specific fold, and it also has a PAM interacting site. And the PAM interacting domain in apoptosis state is very disordered. Only when it's bound with a guide RNA is it active and it's able to recognize the target site. In apoptosis state, the Cas9 binds to DNA non-specifically. It can rapidly attach to different sequences and detach, but in the presence of guide RNA, it can actually attach the specific DNA target sequence and actually make a cut. Mutating one of these domains, the RUVC or the H&H, can give you a Cas9 nickase. Mutating both of these domains creates a dead Cas9. Thank you so much for watching this video. I'll be sending you out new videos this week about the type 1 CRISPR system and the type 2 CRISPR system. I hope you're enjoying these videos. The videos I'll be sending out this week are going to go much more in depth. This video was intended to be an intro video. I hope you enjoyed this series, and thank you so much for watching. Bye!